Okay, so now we are going to transition to Pete Knox, who is our Senior Director of Research at the LifeRaft Group. He has been a wonderful participant during this New Horizons meeting and other ones in the past, and we're really looking forward to his presentation today. Pete, you can begin.
Thank you, Sarah. You can follow me around and give me an introduction like that any day you want. That's fine. Thank you. And first, I want to thank all the other presenters. It was interesting information. I'll probably be referring in this presentation to some of the things you talked about. And I also want to thank all the other attendees who hung on this long. Okay, we're almost there, folks. If this was the Kentucky Derby, they'd be saying round the stretch they come. So get your mint juleps, hold on, and we'll be done soon, but it's going to be worth some information. So let me just share my screen here and we'll get going, okay? And I'll explain what it is I'm talking about. Hold on a second. Can everybody see that? Anybody see that? Okay.
Yes.
Okay. All right. So what this talk is about, and it's going to draw on a bunch of studies that have already been done, and I'll talk about that, has to do with placebos, okay, and alternative clinical trial designs. It also has to do with real-world data and real-world evidence, which is something that's very important, not just to the LifeRaft group, but in general now in this field. And not everybody necessarily knows what it is. I'm not going to assume that. So that's one of the first things I'm going to do is define that. So what is real-world data and real-world evidence? Okay, it's really a definition in many ways of exclusion by what it isn't. If you think of clinical trials and the information collected in clinical trials, this is not that. It is the other stuff. So it is the information that's collected outside of that formal process. It could be surveys, it could be registries. You know, there are medical devices now that collect, including your phone, that collect information. That's all real-world data. That data, when you then take it and make some conclusions based on it, report some statistics based on what you found, that is now real-world evidence. So in order to have real-world evidence, you have to have real-world data, okay? But that's great, but what does that mean in terms of other data? Well, we've never said it is, but we have to point out that real-world data and the evidence associated with it is not adversarial to what is collected in traditional clinical trial process. It is complementary, okay? It's designed to supplement and not replace. Really what this is being able to see the entire picture of what's happening, especially the patient perspective, which those of you who participate in clinical trials and some of you researchers who are honest, who are being honest, know is sometimes excluded or not always easy to find. So it's something that is emerging, partly due to regulatory concerns like the 21st Century Cures Act. And again, like anything that's emerging, it's an in process. There are groups like ISFOR and other groups who are really trying to nail down what the standards are for real-world data and real-world evidence, okay? But this is about placebos, right? So what does it have to do with placebos, real-world data and real-world evidence? Well, again, this is supplemental information that can be used to understand stuff. We have known at the LifeRaft Group for years, we believed, I should say, that patients who are in the placebo arm of a trial generally didn't do very well. We're talking about the trials where the drug is good, okay? And we'll look at some specific trials in a moment. So we started collecting information on progression-free survival and overall survival in our patient registry. We've been doing that for many years. And many of those participants were in clinical trials. So that's the real-world data we were collecting to help see if it could provide real-world evidence to solve our hypothesis of does this harm patients. So we combined that info with information that came from actual clinical trials. So see how it's complementary to see, is this really what's happening? Is it harming patients? That's the big question. Is it harming patients? Okay. So instead of just answering that question off the cuff, I'm going to take a look at the data from a number of important clinical trials, many of which we've already mentioned. Trials where we're going to have information about imatinib and sunitinib and regorafenib and ripretinib and even pemetispib. We're going to talk about that in our very wonderfully written publication, LRG Science. So look, this is just about all of the real medications, but wait a minute, not all of them. Isn't there one missing? Hold your horses, Kentucky Derby. What about Avakid? What about avapritinib? Hold on. No, it's not in there. Why not? Because that one didn't use a placebo. Okay, it was the DA4TV trial that Suzanne George referred to. I think that was either yesterday or today. It's getting to be a blur. But not only did it work, we were able to do that trial, but remember that drug got approved So this is possible. It can be done. I'm not saying it's easy, but it's possible. Kudos to Blueprint for taking that risk. Okay. So I'm going to show you, I'm going to go to another screen right now. I'm going to show you an article that was recently published in LRG Science, which is the official science publication of the LifeRaft Group, and you can find anytime at that URL. And I'll show it to you right now. So hold on one second. And it's a large article. And I also want to point out up front, I'm going to refer to some clinical trials, and I just want to be fair. I'm not picking on people with this, and that's not my intention. Okay. I'm just going to lay out information that I'm not the first and we are not the first to say, but let's talk about it. So this was a huge issue that talked about placebos and whether or not it's time, remember that phrase, it's time to rethink placebos, written by Jerry Call. So I just want to highlight some information. Kinloch, which we've all heard about today, ripretinib, was a trial. And if you heard me speak, I believe that was yesterday, to the gentleman from Montreal, I asked him about the placebo effect in that trial. And I think now you guys know why I did that. So there was some demonstrable effect that perhaps that placebo may have harmed some patients. It was a placebo trial with crossover. And a number of patients were not able to cross over, okay? And if you don't believe me, and you shouldn't, you can believe Jean-Yves Blais, you can believe Serrano and other folks who said the rapid clinical decline in patients who received placebo, which might have prevented approximately a third of patients from crossing over to the ripretinib group, highlights the need for new treatments and might ultimately limit the use of placebo controls in future studies evaluating treatments that are equal or superior to fourth-line therapies in gastrointestinal stromal tumor. I'm going to say up front, I'm not saying every single trial out there should not have a placebo. I'm actually going to give you some information about an organization that has come up with kind of a rubric to determine whether or not clinical trial with a non-placebo alternative, which we'll kind of talk about, is a benefit. So I suggest you guys read this article, but let me also show you some data. So this is a result of a bunch of trials, okay? And I'll summarize it a little bit, but again, all the trials we mentioned, not Ayvakit, are on here. So you have sunitinib, you have regorafenib, you have ripretinib, Kinloch. You even have an imatinib rechallenge. Remember Dr. Kang talking about that trial? Okay, right here. And then you have the pimetispib trial in Japan, okay? Now I want you to notice a couple things. I want you to notice that if you take a look at the median progression-free survival. Okay, you don't need to be a statistician to see this. Look at the difference. That's a pretty manageable difference, and your p-value will tell you that's a statistically significant difference. In all cases, these statistically significant differences, not surprising considering these drugs, now pimetispib is still kind of going, these drugs have been approved, so of course they're good results. So that's one thing to notice. The other thing to notice is that look at the results in the placebo arm. 1.4 months, 0.9 months, 1 month, 0.9 months, 1.4 months. They're remarkably consistent, okay? So what we're kind of saying is, gee, patients who don't take anything kind of respond the same. Again, probably not earth-shattering to people, but it's worth noting, and you'll see why in a moment. So we have a demonstrable difference between placebo and non-placebo in the good trials, and we have consistency in the placebo arms. And that's very important. And here's just a graphical version if you want to see it, okay? Here's all the folks who are on placebos. Here's all the folks who are not on placebos, okay? You don't see p-values on this chart, but if you go back to these charts, you see all these p-values are basically ridiculously significant. So that means, commutative property math, that these are also significant, okay? So there's a significant difference. That's important, and I'll get to why in a moment. The other thing is you say, but how many patients are there? Well, let's take a look. If you look at these trials, the sunitinib trial, these are your placebo patients, had about 100 patients. The grid regorafenib trial, depending on which what you're looking at, there was 39 or 27. So even if you took 39, that's fine, but you can add that up, and that's what, 66. Same thing over here for the Deciphers Invictus ripretinib trial. That adds up to 44. The imatinib challenge in Korea is 41, and then the Japanese trial is 28. So there are patients that are out there. Now, why is he going off about all of these points? Because an organization called CITEL, which we became aware of due to our membership in CTTI, the Clinical Trial Transformations Institute, who's looking at trying to make trials better, has come up with the idea of a synthetic control arm. So what's a synthetic control arm? It's basically taking historic placebo data and reusing it. So we had a placebo arm, we don't have to do it again, okay? Now, how does this work? Does it work for every trial? Absolutely not. But there are some requirements, and here they are. If you look on the left, you'll see the requirements, and you can look ahead if you like, but I'll read it to you. Look on the right, you see why GIST is the perfect model for this. So is the patient population molecularly defined? I think we can agree that GIST is pretty molecularly defined, if not the most molecularly defined disease in all of oncology. Are there clearly defined historical or real-world control groups? So in other words, are there placebo groups? Well, there they are. You can't see me pointing, but there they are. Okay, so yes. Are there ethical reasons not to enroll patients into a control arm? Yes. As we mentioned, patients are not able to cross over in some cases and are being harmed. And going back, I noted that there's significant difference in the placebo versus the non-placebo arm. So yes, the answer to that one is yes. And then finally, have the clinical outcomes been measured by other trials? Yes, we have seen time to progression and progression-free survival well documented and measured across many trials. So really, in terms of, if you were to come up with the perfect storm of a disease to use synthetic control arms, GIST is basically it. So we're kind of advocating for the idea of synthetic control arms in GIST, okay? Now, look, this is a concept. There are some things that need to be ironed out. So like anything else, it's usually not the technology that's the issue. It's often not even the data itself, okay? It's the mechanics, and in some cases, the ethics behind it. So we have to figure out who owns the data. And owning is maybe not a great word, but curates the data. Who curates the data? Because where are we going to get it from? Okay. Are we going to be able to put this data in a public place? Because remember, if you're going back to historical data, you're going to be able to go get it somewhere. And that sounds trivial, but it's not, because there's a lot of trials where that's not the case. And can we run simulations or validations before the next trial using this? What that really is a timing issue. Will doing this mathematical gymnastics, as somebody put it earlier today, will that hold up the trial process? We don't think it will. But these are questions that do have to be addressed. And I'll tell you that there are a lot of groups out there, including the FDA, who are looking at assembling data in arms like this. So I actually think we're going to see that, okay? And as I said here, it's time. It's also time to go back to the slides. So hold on, let me just go back to here. And so we saw the data. So let me just summarize what I showed you, because that was pretty quick. So what do we know? We know placebos in advanced GIST can shorten lives. We've seen that. Even in trials like the very good ripretinib trial that was approved with crossover designs. We've seen that. So not picking on that trial, but there are other trials too where, you know, perhaps being in the placebo was not a good thing. We know that PFS has become if not the primary outcome measure in GIST, because, you know, overall survival takes a long time. It's a good problem to have, but it means the trial takes forever. So that's important, again, going back with Cytel's list. We also know, as I've demonstrated, that the placebo arms in multiple trials have shown very similar results. So really reproducing the trial to get another 0.9 to 1.4 month interval, really it's of questionable value. And now we know, and I've shown you, that there are new trial designs. Now, that's not the only thing, synthetic control arms. There also are multiple comparison arms where you go and you say, all right, I have one placebo. And maybe that's what's needed when you can't hit all of those requirements for Cytel. One placebo, a bunch, you know, like a basket trial, a bunch of different drug approaches, and we'll use that. Hey, at least then you don't have, you know, 4 placebo arms with 4 drugs, you have 1. So that's a nice compromise if it doesn't work. But if you meet those requirements from Cytel, which again is not just Cytel, CTTI is driving that as well, so it'll probably become a standard. That's actually what we would prefer. So we're all basically saying now it's time. You keep hearing me say that. It's time. It's time for new trial designs. You know, and remember, real-world data and real-world evidence can help with this, because these trial designs, when you get this data, this retrospective data can come from things like registries. The FDA has been very amenable to doing long-term studies with registries and looking at registries. So that's an option. So real-world data and real-world evidence is important. And if anything, whatever trial you're doing, even if you don't have a placebo, using real-world data and real-world evidence to supplement the trial information is seeing the whole picture. So it's worth doing. So it's time. We need either common placebo data or a synthetic control arm. We need RWE in those trials. Okay, and the remarkable consistency of our placebo arms has made that possible. And real world data, I'll also point out, being involved in it, it literally gets better every day. It's hard to keep up. So that's a good thing. So there are folks who still question the efficacy and the reliability and validity of real world data, but I would say that that's getting less and less questioned every day as it becomes more of a reality. So again, it's time. It's time for synthetic control arms. It's time to get near the end of New Horizons now, and it's time for questions. So go right ahead.
Okay, we're opening up the floor.
And I can't see you, so should I stop sharing?
Yes, please do.
Okay, that's good. Go for it.
Any comments or questions from participants?
If you're tired, it's understood. Ooh.
I can ask a question. My name is Manish. I'm from Desmoid Tumor Research Foundation.
Oh, Manish.
Hi. And I think this is extremely interesting. I think we all agree that if placebos aren't working, we shouldn't be using them. But how do we get the pharma companies, the companies doing the trials, to release that data to be able to pull them together for the virtual arms?
Yeah, that's one of the questions I point out. It's not just getting them to give it up, it's also dealing with ownership. Because one of the issues is, in some cases, you're talking about trials that have been approved. I was able to get that data because it's publicly available. It was in the trial. But really what you're kind of asking is, what about the trials that never made it? Okay, what about that information? Well, that's challenging. The reality is, in a lot of cases, there's enough trials that made it that you really don't have to deal with that, but that's going to have to come from regulators, and regulators are beginning to understand that. What's really interesting about it, and I won't say who I spoke to because I probably signed an NDA, to be honest with you, but when I first talked about this to a pharma company, unsolicited reaction, I didn't say this, they go, that's interesting. You all heard about, I can't remember who said it, how the biggest problem with clinical trials is they can't recruit, okay? Well, remember, you're recruiting for a placebo arm too. And the person actually said, this might actually help the trials recruit faster and be cheaper. And I went, oh yeah, I think it will. So actually pharma may turn around and decide they want this at some point. So yeah. Do you want me to just read the questions, or are you gonna screen them for me, Sarah? Whatever you want.
Oh, go ahead, Pete. Somebody
Yeah,
just asked a question.
Okay. Thank you, Daniel. Yes, interesting. And I want to thank Jerry Call for doing some of this work too. It was very interesting. So synthetic control arm, a fascinating, hold on, first one. Can trial volunteers condition their participation on not getting a placebo to that help encourage using existing results instead, sort of going on strike? I would think that if you conditioned patients' results going on a placebo, none of them would go on a placebo. I don't think very few people would willingly go on a placebo. And the whole point of placebos in many cases is you do not know, because once you know that you're on a placebo, then the possibility is you're going to say, well, I'm just not going to take the drug, and you're going to have a differentiation. So the whole point of placebos is to not have that. They're generally double-blinded, not always, but not only do you as the patient not know, but often the physician who's putting on the trial doesn't know either, so that you can't. But that in itself, think about that, what you're saying. If trial volunteers are saying, I would only participate in this if I didn't have a placebo, that means there's an ethical concern about placebos. So that in itself says we shouldn't have them, which is why we're doing this. The next question, synthetic control arm, a fascinating idea. I will pass that on to Saitel. Does it matter that combining historical trials for the control is combining patients with different histories, some in first line, some second, some third? You would want to make sure when you do that selection, you have similar placebo arms. That's what's important about it. And I didn't cover everything, but if you're doing a third line study for just, let's say, you would go looking for third line placebo data. There's been placebo data in almost every trial we've ever done. In some cases, maybe you can't get that, but then you would have to look and see, based on the unique design of your therapy and your trial, it may not matter. So it depends, because in some cases they're multi-line trials, so it may not matter. You would just probably want to get a mix that's similar to your response arm. But yeah, I mean, look, unfortunately or fortunately, it's not like we have a dearth of placebo data for GIST. There's plenty of it, so we could definitely get it. Any other questions? Ooh.
Pete, I have a question.
Okay. Go for it, Norman. I'll do my best.
It's really a comment. In the last year or so, we've been working with a couple of key people, including a guy named Greg Simon, who was the head of Vice President Biden's Cancer Initiative. And we have a draft of a protocol, and I've discussed it with a few people in the last week or two with regard to placebos. One of which is to ask a pharmaceutical company or anybody who is sponsoring a trial that if they intend to use a placebo, to come up with an impact statement before the trial starts as to what the consequence of the placebo would be. Very similar to an environmental impact statement where you want to build, it might be a good thing, you want to build a new sewer system, a new water system, what have you. What is your impact on the environment of doing it? And the second piece that we've put into this draft proposal that perhaps people would comment on would be that if a patient and a patient's family suffer unduly from a placebo, that the cost of that phenomenon be factored into the cost of developing the drug. When we had a trial recently where we had an excess death rate of 64% in the placebo group, it's not just a percentage. It's families that no longer have a father or a mother or people who can provide an income for them. And so to take the concept of placebo and take it out of the back room, if you will, and put it up front and let it compete with reality in terms of what are the consequences and who's going to pay for the consequences.
Norman, totally agree. There's actually a comment that echoes what you're saying, but one of the things I'll say is what's funny to me is in some cases, pharma agrees with us and says, yeah, you're right. Maybe, because again, it's going to turn around. And maybe what will happen is, I hope no one ever has to write that environmental impact statement because they won't, they'll just go, we'll just won't use the placebo at all. I think that in addition to the environmental impact statement, if you have an agreed upon, and this was what will happen, if you have an agreed upon standard like what Cytel has, and you meet those requirements, now you're going to have to say, look, even though you meet those requirements for not doing placebo, you have to tell us why you want to do it. The question that's kind of related is, are there not interim consistent analysis to ensure that the control arm is not harming patients? I feel like I've heard of trials that end early because it's abundantly clear the test arm is so much better. Results of the trial you should indicate is with case. Well, so that's kind of two questions. Are there interim consistent analyses to ensure the control arm is not harming patients? I think really what they're showing is that the stimulus arm is benefiting patients to a higher degree than in the placebo arm. And I know that sounds like a statistician's answer, but it's not that it's harming patients, it's that the patients aren't getting the benefit that they should, so they allow for crossover. But keep in mind, the ripretinib study we just highlighted, that's great if you can make it to crossover. So the whole point is we shouldn't be doing that in the first place. So when you say, I've heard trials end early because it's abundantly clear the test arm is much better. Yes, often they include crossover first, but then often, look, not every trial has to go to phase 4. You saw some examples this morning of trials that were approved in phase 1 or phase 2 because it became abundantly clear that it's a benefit. So that kind of ties to what you're saying, Norman, a little bit. But before I answer any of the questions, if anyone wants to comment on what Norman said, go for it. Okay, all right, so another question, which is probably related as well. What is the FDA's role in stopping the use of placebos? That's a very interesting question. I've been in the room with FDA, I've been in the room with pharma, sometimes we're even in the same room, and you get a lot of this. Well, they said this, but they said this. So what happens is it's about FDA saying it's okay. That's one of the reasons I highlighted the AYVAKIT trial. It can be done. So that's FDA's way of saying, look, you don't need to have placebo. Now, that being said, pharma companies will say, well, but it'll make it harder to be approved. Well, let's see. If FDA gets to the point where they look at a synthetic control arm's placebo group as equivalent to a live, selected placebo group, then we're there. My guess is a few courageous companies will do this. And then as that gets forward, then like everything else in pharma, other companies will go, okay, and they'll follow. And then we'll get to a point where it's moot and we won't have to worry about it anymore. That's my hope, at least. Questions, comments, concerns? It's good that there's questions. That's good. How long do I anticipate that taking? My crystal ball's in the shop. Let me see. With the FDA is a little busy right now with a couple other things going on in the world, so it might take a little longer than we anticipated, COVID, but I'm hoping not that long. That's not really the question. The question is, how long will it take until a pharma company gets courageous enough to try it? And the answer is that's already happened. Beta Kid did it, Blueprint did it. So perhaps others will do it, okay? And it's and if they do it, we will feature it in Allergy Science, that's for sure. But that's what it takes. It takes 1 or 2 or 3 groups doing it, and it's happening. And it may not even start in GIST. There may be other groups that do that. The CITEL, I should mention that. The CITEL protocol was not written for GIST. I know it looks like it was because we hit it so perfectly, but it was written for all disease trials. So there's probably going to be a lot of other diseases. If you think about it, we're a rare disease. If you're talking about diseases like breast cancer or lung cancer or colon cancer that have a much larger population set, the likelihood of them having adequate numbers for historical placebos is probably higher. So it might even happen there first, and then it'll trickle down to us. Mary points out that there's so much knowledge here, and we invite those who might be interested in publishing in Allergy Science to contact us. And I echo that, yes. Other comments, questions? Five seconds. To you, Ms. Rothschild, to you.
Oh, well, I think this was a great meeting. I want to thank everybody who stuck with us for 3 days and participated. And we're actually going to be having a summary of the conference in a future report. So we're happy to share that with you. And to thank everybody who's helping to compile that. But I want to also thank the steering committee. That would be David Josephi, Norman Scherzer, Piga Fernandez, Martin Wetstein, Ginger Sawyer. We are actually missing a major person from the steering committee, who is Marcus Wartenberg, who's been with us for many years. But we know that Catherine Schuster and Michi Geisler were here in his place, and we very much miss him. But this is always a group effort in terms of really brainstorming what's needed each year in terms of the discussion and presentations. And we hope that it was to your liking.